Hi, my name is Finn, and this is my Lab 3 video. If you hear a ringing noise in the back, sorry about that. Some idiot set off the fire alarm. Alright, so today I'm going to be reviewing the dendrite area of the neuron, specifically this part right here. And when I say dendrite area, I mean the synapse, how transmission works, and how it relates to action potentials. So, to start off, here is a diagram of what the synapse looks like. So right here we have the terminal button that is basically the end of the neuron that's sending a signal, and the dendrite of the receiving neuron. A few things I need to point out are the presynaptic membrane, which is a part of the terminal button that a neurotransmitter needs to flow through in order to get into the synapse and transmission for to occur. We also have the postsynaptic membrane, which is the membrane that surrounds the dendrite of the receiving neuron, that neurotransmitters don't necessarily have to go through, but they do contain very important ion channels that I will get into later. Another thing I need to point out are the synaptic ventricles right here. Here's a bigger version. The synaptic ventricles basically act as a storage station for neurotransmitters. When transmission is not in effect, neurotransmitters are stored inside the synaptic ventricle. So, in regards to transmission, there are about 4 steps. Step 1 is an action potential signals to the terminal buttons that transmission needs to occur. And the first thing that they do is that they open these gates right here to allow calcium ions to flow into the cell. After that, the calcium ions that just entered will sort of corral the ventricles toward the membrane, where they will dump their contents into the synapse, allowing for transmission to occur. This process is also called exocytosis. The next step involves the actual communication from one neuron to another. This occurs when the neurotransmitters cross the synaptic cleft, this gap right here, and bind to these receptor sites, which allow for sodium ions to flow through and into the dendrite. Once that is done and the neurotransmitters have binded with the receiving neuron, they are signaled to leave the synapse because the following steps cannot occur if the neurotransmitters are still inside. But the question is, where do they go? Well, for that we need to talk about inactivation. There are a few different methods for inactivation, one of which is diffusing, which is just where a neurotransmitter breaks down on its own. Another is reuptake, in which a neurotransmitter is recycled back into the terminal button, processed through the presynaptic membrane, and is reused at other synaptic transmissions. The last form of inactivation is called enzymatic deactivation, in which an enzyme, independent of the whole transmission process, comes in to break down the neurotransmitter into separate parts. Now we get to talk about receptors. Yay! So there are 2 types of receptors. We have ionotropic and metabotropic. Metabotropic is essentially a basic receptor. They have a normal bonding site. They can bond to any type of neurotransmitter. They don't have to have a specific one. They don't have an ion channel, which means they cannot allow ions to flow into or out of the specific part of the cell. They're good for slower responses, and instead of relying on ions, what they rely on instead is chain reactions and G proteins. using G proteins and second messengers. Inotropic, on the other hand, is a bit more like a lock that needs a specific key to open. It can only bond with specific neurotransmitters. Other ones it will not allow. It does have an ion channel, so the process of actually receiving the signal is a lot simpler. It can just allow the ions to flow through the channel. And this is good for quick responses with no after effects. When a neurotransmitter and a receptor react with each other, they can produce two types of reactions. Excitation, which is basically ions flowing into the cell, making it more positive and depolarizing it in the process, which increases the likelihood that it will meet the threshold of excitation. The other reaction is inhibition, which is just the opposite effect. It decreases the likelihood of an action potential by allowing negative ions to flow into the cell and hyperpolarizing it. When these reactions occur, they produce two different kinds of potentials: excitatory postsynaptic potentials, or EPSP, and inhibitory postsynaptic potentials, IPSP. These are basically the two parts that dictate whether or not a reaction is going to, an action potential is going to be reached. But how does that actually work? Well, this all culminates in what's called the axon hillock, the place where the axon meets the cell body of the neuron. How this occurs is the EPSPs and IPSPs are added together in the axon hillock to determine whether or not a reaction is going to occur. Like if you have a +3 and a -1, you have more positives, therefore the number's positive. And how this relates is basically how action potentials are determined. But there's two different methods of adding them up. There is temporal summation and spatial summation. With temporal summation, what that looks like is it adds up potentials that repeatedly occur on only one place in the dendrite. So you'll see right here, the potentials flow into the dendrite through this specific spot and travel from the hillock down to the rest of the body. Another way is with spatial summation, which adds up potentials that occur all over the dendrite, like at all different places, but they have to be occurring at the same time. And then they're added in the hillock and pushed to the rest of the body. And basically how this differs in the kinds of potentials, the kind of action potentials that occur, is with spatial processing. Since they're taking in all different kinds of signals from all different places in the neuron, they can actually reach that threshold of excitation a lot faster than if you were using temporal. Whereas temporal, it takes a little bit longer since they're getting individual signals from only a single place in the neuron. And with that, my video is finished. Thank you so much for watching. And yeah, I know this is like 3 days late, but this took a whole lot longer than I thought it would because I wanted to be creative. So yes, thank you for watching.